Hi everyone. We're gonna go ahead and get started. Good morning and good afternoon for everyone, and welcome to our last day of the symposium. For those who I haven't met, my name is Lucia Lopez, and I work with Global Network here at LSU. Today we will be discussing the topic of food safety, followed by our wrap-up roundtable discussion. I would like to start first by introducing our moderator today, Dr. Ariana from our School of Nutrition and Food Science here at LSU. He specializes in enhancing probiotic characteristics, functional dairy foods, dairy food microstructure, and dairy ingredient functionality. Just as a reminder, if you have any questions throughout, just make sure that you put them in the chat and we will be responding them. Without further ado, Dr. Ariana, you can go ahead and start. Thank you.
Thank you so much. I just want to make sure if y'all can see the screen. Can y'all see my screen? Good morning, everybody, and welcome to my presentation this morning on the viruses in the foods and the influence of the coronavirus on the food industry. Next slide, please. Now, virus is a submicroscopic infectious agent that replicates only inside a living cell of an organism. Means you got to have a living host for the virus to make more copies of itself. Now, the size of the virus is around 25 to 250 nanometers. And those transmissible through foods, they are 25 to 30 nanometers, and some as large as 75 nanometers. What does that mean? Like, to some, these are just numbers. To put this in perspective, like if a human being were the size of the earth, the virus would be the size of a mouse. Means what the size of the mouse is to the earth, that would be the human being. That would be the virus to the human being. Now, viruses, the chemical composition, it is composed of either DNA or RNA, and then it has a coat around it. It has a protein coat, and it can also have a lipid around it, and it is inert, meaning it cannot reproduce by itself. Okay, and autonomously, it needs a host to make copies of itself. Next slide, please. Okay, now the bacteriophage lytic cycle. Now, we were just talking about how a virus needs a living host to make copies of itself. Now, if you look at if this is the bacterium, if this is your bacterial culture, like in the cheese industry, and we basically added the bacterial culture to the warm milk, okay, and if there is virus around, the bacteriophage, it's a virus that attacks bacteria, and first what the virus will do, it first needs to attach to the bacterial cell. Once it attaches onto the bacterial cell, then it will pass on its genetic material into the bacterial cell and take over the bacterial cell machinery. Then what happens is it makes copies of its proteins, the protein coat, and it makes copies of its genetic material, the DNA or the RNA. And then once it makes a lot of copies of it, then there is the assembly stage in which the genetic material is packed into the protein coat, and more copies of viruses are made within the bacterial cell. Then it comes to such a stage where it cannot take any more copies, and the cell lyses, and you have many more copies of the virus. Just to give you a sense of how many copies it can make, like if the milk is warm when you added your bacterial culture, in 1 hour, if your bacteria goes from 1 cell to 2 cells, and then 2 to 4 cells, the virus has gone from 1 copy to 150 copies. Means the time it takes for the bacterium to go from 1 cell to 2 cells, the virus has gone from 1 copy to 150 copies. and then the time it takes for the bacterium to go from the 2 cells to the 4 cells, the virus has made another 150 copies times 150 copies. Means by the time the bacteria from 1 cell has gone to 4 cells, the virus has 150 times 150, that is 22,500 copies. So that is just to let you know how fast it can replicate within the host cell if the conditions are right. Next slide, please. Okay, next slide, please. We're talking now as food as a vehicle for transmission for viruses. Now, the polio virus, the human disease poliomyelitis. Now that was first reported in around 1914 to 1949, when this virus, the polio virus, was transmitted via the raw milk, and it was transmitted through the fecal oral route. Now we will see this term a lot. Excuse me, fecal oral route in terms of virus transmissions, and that basically just means that someone has not washed their hands and touched the food, prepared the food, an infected person who's not washed their hands and prepared the food. The virus multiplies in the intestine, and the virus invades the nervous system and can vaccines, improve sanitation, and increase use of pasteurized milk. Now you see that date out there, 1949. So basically, then the importance of pasteurization in milk was known more and more. And then people started to drink less of raw milk and more pasteurized products, pasteurized milk. Next slide, please. Another virus, the hepatitis A, it is the infectious virus. This also is transmitted via the fecal oral route. Now, the incubation period is about 15 to 50 days, and the virus shedding begins 10 to 14 days before the onset of the symptoms. Means you don't even see the symptoms yet, and the person is already shedding viruses. Okay, so that adds more to that fecal oral route we are talking about. And the virus infection, it infects the liver and sometimes causes jaundice. Now, the shellfish are often served as a vehicle because of the waters in which they are grown are subject to human fecal pollution. Just dumping of fecal material sometimes by boats and by other ways. Virus collects within the shellfish because of the filter feeding method. The humans eat the virus containing the digestive tracts of the shellfish along with the rest of the animal, and the shellfish is often eaten raw or undercooked, and that is why the virus stays alive within the shellfish. If it were heated in some way, that would take care of the virus. So our shellfish appears to protect the virus during the heating. It has like a protective effect. The foods, the salads, the sandwiches infected by infected humans serve as a vehicle for the hepatitis virus. Now, if you see the salads and sandwiches, that they basically do not go through heating before you eat it. So there is no kill step before you eat it. So if an infected human being has handled those foods and served them, so that's how it gets in through the fecal oral route. Next slide, please. The Norwalk-like virus. Now, it was first reported as an outbreak of the gastroenteritis in Norwalk, Ohio in 1972. Now, here, this is the diarrhea and the vomiting. And this also is a very small virus compared to some of the others. It's 32 nanometers. And this, again, is the fecal oral route, okay? The primary transmission mode. It can be airborne, and even by transmission through like objects like doorknobs, faucets, things like that can help spread the virus. Any food item can potentially be infected with the Norwalk-like virus through the fecal contamination. Again, you're looking at it fecal, because the virus stays in the GI tract, and it can, and when the feces sheds in the feces. Now, certain foods are implicated more often than others. Again, we are shellfish, back to shellfish. And it prevents the contamination preventing the contamination of the in-harvest waters by human waste, restricting boaters from dumping their waste overboard, are useful means of preventing the shellfish-associated Norwalk-like virus gastroenteritis outbreaks. Infected food handlers contaminating the food is another frequent source. And because of the low infectious dose, a high concentration of virus in the stools, even a limited contamination can result in substantial outbreaks. Again, here it's ready-to-eat foods. Again, why? Because there is no heating step involved and required handling and no subsequent cooking. Again, like salads, deli sandwiches, handled by infected people, infected handlers, and served by infected people. Gastroenteritis outbreaks have been associated with sources of contaminated waters, including municipal waters, well waters, stream waters, and even commercial ice. Now, ice made out of contaminated water would be also a potential risk. Now, if drinking water or recreational water is suspected as being an outbreak source, chlorination is a method. Like about 10 ppm, 10 parts per million of less of chlorine might help in the disinfection process. And frequent hand washing with soap is an effective means. And prevention is reducing human waste contamination of water supplies. Next slide, please. We have rotavirus. This is a fairly large virus compared to the one we saw earlier. It's about more than twice its size. It's present in the infected person's stools. Again, we're talking about stool. Again, it's the infected food handler. Again, it's the fecal oral route. One thing to keep in mind with these viruses with foods, a lot of them, it's this fecal oral route. And some of the symptoms are diarrhea, fever, vomiting, abdominal pain. And treatment includes intake of fluids because you have diarrhea. You need to replace some of those electrolytes. Intake of fluids, good hygiene, washing hands, and vaccinations. Next slide, please. Now, echovirus, the ECHO stands for enteric cytopathic human orphan virus. It lives in the GI tract, and the infection by contact again with fecal material is contaminated by the virus, touching surfaces contaminated with the virus, breathing in infected airborne particles. Again, it's the infected food handler, fecal oral route, and a report way back in 1976 where coleslaw was eaten by 80 people at a picnic in Pennsylvania and showed these viral symptoms. Next slide, please. Now, here we are looking at viruses which are not primarily of human origin that occur in foods. Now, this is tick-borne encephalitis, which is caused by the enveloped RNA virus. Now, these ticks, they bite the cattle, goat, and they get infected, and the virus is shed in their milk. Consumption of raw milk and raw milk products in certain countries has resulted in this tick-borne encephalitis. Now, encephalitis is the inflammation of the brain. Next slide, please. Then again, we are looking at viruses not primarily of human origin that occur in foods. These are the oncogenic viruses. These are the viruses that cause cancer, tumors. The avian leukosis virus causes tumors in chickens, present in poultry carcasses and in eggs. You have the bovine leukosis virus, causes tumors in cattle. The virus occurs latently in lymphocytes in the cattle carcasses and in milk. We have the polyoma virus. It causes tumors in rodents. It is shed in the urine of rodents and has been detected in stored grains where rodents were many. Next slide, please. Now, contamination of food. The primary contamination is raw material. It occurs during harvest, slaughter, or either you have the infected animal. And during harvest, it is like sewage. We talked about how it's fecal contamination. It is a sewage in the irrigation waters. A secondary contamination is during processing, storage, distribution, or final preparation of the food. You can have it indirect or direct. The indirect is when polluted water carries the virus to the food or food contact surfaces. And you can have it also through vectors such as insects or rodents can carry the viruses. Now you have a direct contamination. That is by infected food handlers. Most common way of shedding viruses in the feces. infected the amateurs handling the foods. So again, this is we're talking about the fecal oral route again. Next slide, please. Inactivation in the food for the virus to lose its infectivity. Now, viruses cannot multiply in foods. They need a living host. Virus stability. The virus persists in refrigerated or frozen foods, but is rapidly inactivated at cooking temperatures. That cooking, that heating step is very, very critical. No virus is indefinitely stable at room temperature or below. The physical inactivation by heat, like pasteurization, affects the virus, nucleic acids, and the protein coat. Like heating would affect proteins, right? And at some temperatures would denature proteins. And so that is how it basically is effective against the virus too. So, and drying inactivates some viruses. Now, this drying is not by like spray drying or roller drying where you use heat. Experiments were conducted where freeze drying was used. Now, no heat, it's just freeze drying. And there was 99% inactivation of experimentally added viruses during the freeze drying process, freeze drying of foods. Then irradiation is another way. The UV rays from sunlight or germicidal lamps are quite effective against viruses on the surface exposed directly to irradiation. You have gamma rays from cobalt-60 sources capable of penetrating into the food and inactivating the viruses. 90% virus inactivation by dose of 40 kilogray, 0.43 megarads. Microwaves are also are effective against inactivating viruses. Next slide, please. Now, chemical inactivation, we looked at physical methods earlier. There are chemical methods too. And now we are talking about whether the food, whether it's on the surface of the food, viruses on the surface of the food or within the food. Now, on the surface of the food, disinfectants, generally strong oxidizing agents like ozone in the water or chlorine, are active in the viruses in the water on the surface of the foods. And chlorine acts most rapidly when in the form of undissociated hypochlorous acid, and more slowly as the combined chlorine. But other chemicals, agents such as phenolics and quaternary ammonium compounds are less effective against the virus as compared to chlorine. Now, if the virus is within the food, externally the disinfectants will not be able to get it. It's within the food, right? So some viruses are inactivated by sodium bisulfide. And there's the fat and the protein of the food tends to have a protective effect on the virus, while acidity of the food tends to have a retarding effect on the virus. So pH of the food is important. Next slide, please. Now we are on the second part of the presentation, is influence of coronavirus on the food industry. In March 2020, because of the shutdown and stay-at-home orders, caused a change in the purchasing behavior. People was basically eating more inside the houses than outside. Now, this not only restaurants were shut down, even theme parks, cruise lines, airlines were shut down during that initial period. And in a matter of days, many farmers lost the entire market channel. Now, we're not dealing with a shortage of food, we're dealing with a surplus of the food. There is excess and there's wastage of the food. Now, the potato farmers, for example, for the French fries, the potatoes which were used for French fries, the restaurants shut down. So there was not as much French fry consumption. And so there was just a lot of surplus of potatoes. And millions of pounds of fresh produce, such as cabbage, green beans, lettuce, destroyed. They were ground up back into the ground as coronavirus disrupted the food supply chain. Here we see basically how the tract is basically grinding up the produce in the farm itself, because there is just no market for it. All the restaurants are shut down, and they just basically, if you think something like lettuce, what's the shelf life on it? It's about like 2 weeks, right? So, and they don't have place to store all that stuff, and because there is no market for it. Next slide, please. Now, here we are looking at the same story for other commodities too, like eggs and the milk. They also, when they were being produced on a large scale, just on a scale prior to the pandemic, and then the shutdown, there was no market for them. And so hundreds of thousands of eggs were smashed daily. Dairy farmers dumped milk on their fields because there was no one to buy it. Now, prior to the pandemic, about 50% of the milk produced in the USA went to restaurants and other food service operations. These food service operations were like coffee shops, bakeries, things like that. So most of the dairy farms did not have the equipment needed to store the excess capacity safely. Now, milk is the fluid product, it can spoil readily. And it also has a shelf life of like 13 days. So, and there is no place to store so much of fluid product. And the farmers were not, it was not easy for the farmers who typically deliver milk for use in cheese to rapidly move to selling in the grocery stores. Now, in the grocery stores, the story was different because people were eating at home. They would go shop in the grocery stores. But now the farmers who are selling this milk for cheese purposes, they are selling it out in tanker trucks. And now in grocery stores, you need them in these little gallon jugs, in these little containers. So packaging became an issue. And one of the obstacles was rapidly adjusting to the dairy supply chain was the packaging. Equipment and material for different, for packaging, labeling, for commercial food service was different than for grocery stores. Like, for example, if we go to the grocery store and we buy small cups of yogurt. But for example, in restaurants, they basically get yogurts in the large tubs. Like for example, when we buy cheese, we buy grated cheese in small packets. But if it's like a pizza manufacturing facility, they need their shredded mozzarella in large boxes, in like 40-pound boxes. So the whole packaging is different. So basically, they had to change gears to this new kind of packaging now to cater to retail stores versus restaurants, and that was becoming a big issue. Next slide, please. Now, again, retail food service is at the customer level. There was hoarding food items, and in the grocery stores initially there was hoarding of food items. There was shortage on the shelves in grocery stores initially. What was easy to divert? It was not easy to divert all produce intended for restaurants to grocery stores instead, because again, it comes down to this packaging issue, the distribution chain. Like, for example, is veggies. If you buy like a small bag of veggies, like a small bag of peas, for example, restaurant would take it in large containers, large quantities. Packaging was the issue. The second issue was the distribution centers. Now, these distribution centers, if they were at 100% capacity, now with all this, they had to operate like more like at 200% capacity, and they could only operate like about 130 to 140% capacity. So distribution centers, that was becoming another issue. And then a lot of farmers didn't want to basically just waste their crop in the field, so they had to adjust they were actually spending money on harvesting and processing and transport for donations, basically donate all that food and in that process to harvest that crop, they had to incur those costs themselves. Next slide, please. Then the government basically stepped in. Then with aid packages, and in around the mid of April 2020, the White House announced about $19 billion farm relief package, which set aside so many billion in direct payments to farmers and ranchers, and in government purchases of dairy meat and produce intended for distribution to the food banks. The product caps were at 125,000 per product and 20,000 per farmer, but some crops like cabbage took a lot more dollars to grow than soybeans, for example, but all crops had the same ceiling. And the USDA spent several $100 million purchasing excess produce, dairy, and meat, and just to basically turn it around and give it to food banks. These items were combined in a variety of ways in boxes and distributed to food banks and community organizations. Next slide, please. Now, the meat plants. Now, the meat plants is because of the close proximity of the workers working in the meat. There were a lot of workers that were getting infected, and several died. And there was a record number of meat plants over time shut down. And then there was like a shortage, like there was a big concern that there would be a shortage of meat in the country. And so the president signed the executive order that is the defense to invoke the Defense Production Act. Now, what is this act? This act authorizes the president to require companies to prioritize government contracts and orders seen as necessary for the national defense, with the goal of ensuring that the private sector was producing enough goods needed to meet the war efforts and other national emergency. So this made the ensured that the meat plants stay open, and also the order protected the companies from legal liabilities if the workers got sick. And the workers had to stay more spaced out in the plants, and hence that meant is many cattle, as prior to the pandemic, could not get processed. Next slide, please. Now, in relationship to food, according to the CDC, coronavirus in relationship to the food, according to the CDC, the risk of getting sick with COVID-19 from eating or handling food, including frozen food and produce and food packages, is considered very low. Now, coronavirus resides in the body fluids of the mouth and nose cavity of infected people, and it is thought to spread mostly person to person through respiratory droplets when someone coughs, sneezes, or talks. Now, that is the main route of the spread of the virus. Food or food packaging material is not thought to be the main way coronavirus spreads. The virus that causes COVID-19 has not been found in drinking water. The Environmental Protection Agency regulates water treatment plants to ensure that treated water is safe to drink. Coronavirus lives up to 2 to 3 days on plastic containers, cheese bags, and stainless steel food contact surfaces. Next slide, please. Now, in conclusion, a brief one. Cleanliness and disinfection of high-touch surface areas such as handles, kitchen countertops, faucets, light switches, doorknobs is critical, and it is important to safely handle and thoroughly wash, cook foods to the recommended cooking temperatures to inactivate the virus or remove it by washing. That's next slide, please. That's pretty much all I had. Next slide, please. And that's, I just thank you all for your time, and I would be willing to accept questions. Like we've seen in earlier viruses, like how we talked about the fecal oral route, as we've seen for the earlier viruses, like it is, but the spread with coronavirus, it's more like these droplets that come out from during sneezing and coughing and even talking. So, and that's why they recommend like the face mask and 6 feet between people, even workers in plants, such as meat processing plants, and washing hands, that simple washing hands, that water does something to the virus. It kills it. It inactivates it. So its temperature is very important during your washing hands too. Warm. Questions?
We do have a couple of questions on the chat.
Sure.
So the first one is there a virus associated with the consumption of alligator meat, or in the case of other aquatic animals in Louisiana, not fish?
Now, we got to keep in mind contaminated waters is the whole issue. Now, it is again, and cooking is the other issue. Now, just as we've seen for shellfish, now basically if these alligators are harvested in contaminated waters, sewage contaminated waters, you have these boats that catch these alligators, and where do they dump their sewage? They're pretty much all day on the boat. There is no place, where do they dump their sewage? So whether those people are infected or not. If they're not infected, then the question of virus does not come up. But if they are infected and they are shedding viruses in their feces, And if they dump their feces in the waters and those waters are contaminated, and then alligators are in those, if the alligators are in those waters, then that's one chance of getting it. The next chance is basically it's how you basically prep the animal first. Is we looked at earlier direct contact and indirect contact. Is how it is basically how is it washed off properly first, and then it is the cooking step, the kill step. So that would be the same thing would apply for alligator meat too. Did you have another part to that question?
Oh, no, but there is another question.
Yeah,
So
sure,
the
sure.
next question is, what strategies can be used in the future to reduce the need to waste food and milk when supply chains change so drastically, like during COVID?
Yes, that's a very nice question. It is basically in milk, for example, or in lettuce or whatever else. The main issue for spoilage is the water in there. How much of water is there in milk? There's 87% of milk is water. And it's got all these wonderful nutrients in there. You have the lactose in there, you have the proteins in there, you have the vitamins in there. All these things that the microorganisms need for growth are in milk. So basically, you dehydrate the milk. The technology would be drying technology. That would be have like some ways to have dry, some way to dry. In case of lettuce, if it whatever way, it can be basically shredded and dried, or some way, or made a slurry out of it and dried. But long story short is to get the water out of the produce and to convert that into another product, and then use that, like how we have reconstituted milk, we have milk powder, we'll have to do something like that for lettuce. And that would be basically to get the water out. That would be that technology needs to be worked on. Pretty much now for drying, what do we have? We have spray dryers, roller dryers, freeze dryers. And two of them use freeze drying is expensive. So also when you work on these drying technologies, you also got to work on cost. So because that's how the farmer thinks, is how much is it going to cost me to do this operation? How much am I going to come out? How much is going to end up in my pocket? So it is the way I look at it is the drying technology and effective and economical drying technology. In drying, like for roller drying and freeze drying, for roller drying and spray drying, heating is involved, and during that heating process, the virus will get killed. Now, drying means you got to get the water from the liquid state to vapor state. So you have to go above the boiling point of water. At least you're going to be at least at the boiling point of water, or a tad above. So for it to go into vapor phase, and those temperatures inactivate viruses. Other questions?
No, we do not seem to have any more questions as of now.
Okay. Well, thank you all so much. Well, okay, our next speaker is Dr. Achyut Adhikari. Dr. Achyut Adhikari is an associate professor and extension food safety specialist at Louisiana State University Agricultural Center. He is involved in planning, developing, implementing, and evaluating comprehensive issue-based educational outreach programs related to food safety regulations. He works closely with food producers, packers, and processors on issues related to the U.S. Food Safety Modernization Act requirements. Dr. Adhikari is a certified lead trainer for FSMA, that's the Food Safety Modernization Act, FSMA, preventive controls of human foods, foreign supplier verification program, and the Produce Safety Alliance growers training. He assists small and medium-sized producers and processors with good agricultural practices, good handling practices, HACCP, and food safety implementation through workshops and field visits. Dr. Adhikari also manages a research program in the areas of understanding the persistence of foodborne pathogens in food production environments and developing food safety interventions for pathogen risk reduction. His current research focuses on identifying and developing alternative practices to mitigate food for microbial food safety risks during pre- and post-harvest operations. Dr. Adhikari's microbial testing lab provides services to Louisiana industry on environmental monitoring, microbial testing, shelf life, and challenge studies, and developing GMP, that's good manufacturing practices, and HACCP, that is hazard analysis critical control point plan. Dr. Adhikari is the current chair of the multi-state project S1077 Food Safety Risk Analysis. His international collaborations include universities and institutes in Poland, Central and South America, Southeast Asia, and Australia. We have Dr. Adhikari with presenting on the next presentation. Dr. Adhikari, all yours.
Great. Thank you so much, Dr. Ariana, for great introduction. Thank you. Let me pull out my slides and then we'll go from there. Okay. You guys see my slides?
Yes, sir, we sure do.
Awesome, thank you. Good morning and good afternoon, everyone. So today I am going to highlight some of the strategies that we can use to control microbial risk in a food facility. So in today's presentation, I am just going to give you a very brief overview of hygienic zoning and environmental monitoring program for food industries. Okay, so the first step, to minimize microbial risk in a food production environment is to understand, the nature and the type of microorganisms, how these microorganisms will behave in a food facility. Normally, there are two different types of microorganisms that are present in a food facility: transient microorganisms and resident microorganisms. So transient microorganisms are those organisms that are introduced into the facility through several different inputs that we use, like raw materials, like personnel, packaging material, the things that we bring from outside, and the personnel that are working in the facility. And those transient microorganisms are very temporary. They could be removed through normal cleaning and sanitation processes. And they typically do not become established in the environment. But on the other hand, the resident microorganisms, they do become established in the environment. And they persist for a long time. And even that we use the normal cleaning and sanitation process, they may not, they may not eliminate those organisms. They may reduce the number, but may not eliminate those microorganisms. And when we are working on reducing microbial risk, as I said, when we have to understand those microorganisms, we need to really focus on three different categories of organisms. One is the index organisms, indicator organisms, and then comes the pathogens. So index organisms are those organisms that you look for that very closely correlates with the pathogens of interest. For example, if you are looking for a Listeria monocytogenes, then you may be focusing your examination or monitoring for Listeria species. Indicator organisms, on the other hand, will indicate the sanitary conditions, like how your facility sanitary or hygienic condition is. And some of the examples of indicator organisms are like coliforms, generic E. coli. And the negative test for indicator organisms doesn't necessarily indicate that there will be no pathogens. It's just the indication of the probability of the presence of pathogens. So both the index organisms and indicator organisms indicates the risk or the probability of pathogens. So when we talk about pathogens, Dr. Ariana just mentioned so nicely about the viruses. The other pathogens are like parasites and bacteria. And when we talk about bacteria, like bacterial pathogens in a food industries, you may have heard this Salmonella, Listeria, and Coronabacter so many times. Like we have so many different recalls, food infections related to these three organisms. Let's talk about Salmonella. If you have a facility that produces low moisture food, these organisms is of really great interest. And when there is an infection with these organisms, it normally causes diarrhea, fever, and abdominal cramp. Listeria. This is a really interesting organism, Listeria monocytogenes. It is very much interesting because it is an environmental pathogen. So if you go outside, like if you go outside and if you grab a bunch of swab from outside, send it to the lab, there is always a probability that you may get a positive Listeria species or Listeria monocytogenes because it is everywhere in the environment. But the problem with Listeria monocytogenes is 20 to 30%. Like if 5 people are infected with Listeria, there is always a chance that 1 person may die, and it can cause meningitis, pneumonia, and a worsen. The third one is Coronavector species, like especially when you are preparing food for high-risk individual, especially this infant formula, it can cause extremely rare disease in infant. So these 3 organisms are really important, like when you are working on minimizing the microbial risk associated with your food production environment. And when you are working on that area, you also need to focus on the risk associated with the food itself. So these are some of the important points that FDA highlights, like in the definition regarding high-risk food. High-risk foods are those foods, that will provide environment for the growth of pathogenic microorganisms. The intrinsic properties of the intrinsic properties of the food may provide an environment for the growth of organisms. Like if the food has high pH or low acid, then microorganisms may grow. Like if the food has high water activity, then there is a chance that pathogenic microorganisms may grow. The another things to consider when you categorize food as high risk or low risk is there any chances that during the production or packing that the food may come in contact, or there is a risk of contamination? Or is there any steps taken into the processing line that you can minimize those microbial risks? So those things are really important to take into considerations. And in addition to that, is there any likelihood of the infection or foodborne illness related to that food in absence of a control? So those things will define if that food is a high-risk food or low-risk food. And when you are dealing with a high-risk food, one of the important strategies to minimize the microbial risk is hygienic zoning. Simple, like hygienic zoning is you are dividing the facility into based upon different risks. So the whole facilities, are mapped into different risks depending upon the flow of the traffic, depending upon the movement of the personnel, the material, the product, and then the waste. So you are controlling the movement of any risk from the low risk area to the medium risk area to the high risk area. And then through the hygienic zoning, you can minimize the microbial risk. We will discuss about that a little bit more detailed. So you really need to keep into mind, there are different conditions, there are different factors that you really need to keep in mind when you are developing hygienic zoning or environmental monitoring program. One of the important things is the product characteristics. What is the characteristics of the product? As I mentioned just before, is the pH. Is the product has high acidity and low water activity, then there will be a very low chances of growth of any contaminated microorganisms. The product itself does not provide favorable environment. So hygienic zoning or environmental monitoring program is really does not relevant to the food that does not actually provide favorable environment for the growth of pathogens. But if the product or the ingredient there is a potential of contamination or provide favorable environment, then hygienic zoning and environmental monitoring program is really important. The another thing that you really need to take into considerations is, does the product, receive a validated process control designed to kill environmental pathogens? Is there any pasteurization step, any cooking steps that will kill those microorganisms? For example, we can take an example of canning. With the canning, there is a thermal treatment, and any potential contaminant in the food will be killed during the canning process. And the food will not be exposed to the environment after the kill step. So there is very low risk. That's why the hygienic zoning or environmental monitoring program may not be much relevant. But at the same time, if you have a kill step, for example, any ready to serve food, okay, So if you have a kill step and then your product is exposed to the environment during packing, then there is high risk. The product may get contamination from the environment, and in those cases, hygienic zone is very, very important. Another important factors that needs to consider is are ready-to-eat ingredients used to produce a ready-to-eat product. That means that an example is a salad. So you have a salad processing line, you are purchasing salad, different ingredients like cheese, lettuce, meat, like a cooked meat, all those things. Those are ready-to-eat ingredients. You are mixing in your facility, and then you are packing and then selling out. That means you don't have any kill step, and then the product is exposed to the environment. Those are high-risk food, and definitely you need a hygienic zoning and environmental monitoring program. That another one you need to take into considerations is if you have a refrigerated food and that food provides or supports the growth of Listeria monocytogenes. So all those factors need to be taken into considerations when you are actually developing your hygienic zoning or environmental monitoring program. Here are some of the examples how you can actually do the zoning of your different areas of your facilities. For example, here you can see non-manufacturing sites. Okay, the more non-manufacturing sites, you have personal, that's movement over there. You don't want any contaminant, any microbial contaminant that will come from the non-manufacturing site to go to your medium risk to high risk areas like the primary pathogen control, or the medium risk like the transitional areas. The transitional areas are those areas where like you have maybe have a hand washing station or entry rooms, like locker rooms that enter into the basic GMP area. So any contaminant any potential contaminant in non-manufacturing areas and transition area, you can still control by the basic cleaning and sanitation process. But when you are discussing about the raw material receiving storage, and especially like the raw materials, as I mentioned before, if you have raw materials that are ready to eat, you really need to focus on that. Sometimes it can be used by the basic GMP control, or in many cases, like if you're really dealing with high risk food product, then you probably have to focus on sanitary preventive control that target any potential contaminant in that area. When moving to the primary pathogen control area, you have product, like I already go through the kill step, or like a process control, and your product is exposed to the environment. Here, basic sanitation or basic GMP may not be enough to control the risk of cross-contamination. So here you may have to really focus on how you can develop a preventive sanitation program to control any potential contaminants. And again, like when you are working with much more sensitive food, like if you are producing food for high-risk individuals, like infants, like infant food, if you are producing those types of food, you really need to focus on. And then the sanitation preventive control is the only answer for those high-risk areas. This is an example of hygienic zoning map of a facility. So here you can see in like a different areas. So here, this area, okay, sorry, let me get a pin. Okay, so here you have this area of like a low risk areas. You have this another area, low risk areas. When you are developing your program, like for hygienic zoning, you really need to keep in mind the traffic flow from these low risk areas to these basic GMPs areas over here, all those basic GMPs areas, and then preventive controls areas over here. You really need to focus. This is a high risk area where your product is actually exposed to the environment. So here you have this cook area where your product is cooked, and then any potential contaminant on the product is already killed. But once you move this product from here to the assembly wrap area, this product will be exposed to the environment. And you really want to make sure that there are no transient or any other microorganisms, like that are present in that environment that will probably contaminate the product. So, you can control the traffic by, by probably, by putting physical barriers, signage, like a color-coded shoes, color-coded aprons, mask, depending upon the flow of this traffic, depending upon the flow of your material, you can use different strategies in order to control or the risk of cross-contamination. Here are some of the examples of your traffic, like a traffic flow, how you can manage that. So here, if you have your traffic flow or movement of raw materials and then the finished product that comes very closely together, if there is a less proximity of those raw materials and then the finished product, then your program may be very poor. There may be a high risk of cross-contamination. If there is an overlap like this, another one, if you have an overlap of your raw materials and then your finished product going to the same way, then is a really high risk. Like there is a potential of cross-contamination. If you have like a product that goes a little bit further, you have raw materials coming from one side, but the product, your final product, has a distance. Like you have a barriers between your product and then your raw material and your final product. Then that's much more better. If you have just like one-way system, like your raw materials comes from one way and then your finished product goes to another way, that is the best one because there is no interaction. Your final product, like does not come very close proximity to your raw materials. That is the best way to maintain the risk associated with microbial contamination. So environmental monitoring program. When we discuss about the environmental monitoring program, basically an environmental monitoring program includes the pathogens or indicator swabbing as appropriate to detect the risk of microbial contamination in your processing environment. So when you are working in environmental monitoring program, you are focusing on how you can identify those niches, those harbor sites that may be a potential source of microbial contaminants. So you are aggressively working on finding those areas that may contaminate microorganisms to your final product. So some important purpose of environmental monitoring is you can also use to verify the effectiveness of your sanitation program. You have been using a sanitation program and then you realize that, your sanitation program is not working. You may change your sanitation program and you now want to make sure that your sanitation program will work. Environmental monitoring program will be very useful in order to validate and verify your sanitation program. It can also be very useful to verify that your hygienic zoning is working. When you change, you have developed your plan of your movement of the traffic, of the movement of your food materials. You want to verify if that is working or not. That's where you use environmental monitoring program. But environmental monitoring program must be tailored to its facility. You can use, you can use an environmental monitoring program of a different facility, but that would be a baseline. That will give you an idea because your facility have different product, it's a different structure, the flow is different, the raw materials ingredient totally different. So it needs to be tailored to your facility depending upon the risk associated with your processing line, depending upon the risk associated with your ingredient, environment, everything. And when you are working on this environmental monitoring program, you may be looking for sometimes pathogens or sometimes indicator or index microorganisms. It depends, doesn't necessarily have to look for pathogens. You can also look for index organisms or indicator organisms to indicate the risk of pathogens presence. But a useful program delicately tries to find the organisms, the source that niches, those harbor sites that may be a potential source of contamination. I discussed about that before also, you really need to consider what type of food product that you are working with and then what is the target microorganism. If you are working for low moisture food, like peanut butter, or any other low moisture food, like flour, that probably would be used to a cookie dough. Okay, you have a flour production industries, probably goes to a cookie dough production facility. Then that flour is a high risk because it is going to use in a cookie dough, and cookie dough may be potentially used as a raw product. So you really need to focus on what type of food product or what type of organisms you are targeting. For low moisture food, salmonella, and salmonella behaves so differently when the moisture content or the water activity of the food materials is different. The ready-to-eat refrigerated food, if you are dealing with RTE food, ready-to-serve food, Listeria monocytogenes or Listeria species, very, very important. And then again, we discussed about that. Infant food, if you are dealing with the infant food, Corynebacter, It's very much important. So depending upon the type of the food material, depending upon the risk, the microbial risk, your environmental monitoring program may target different organisms. And the frequency of the sampling, again, depending upon the risk, associated with the site, when you have those hygienic zoning, you have those different sites. Depending upon the risk associated with each site, your number of samples will be different. For non-manufacturing areas, you will probably collect less frequently and then fewer samples. But when you move to basic GMP area and transition area, and actually to the pathogen, primary pathogen control area, you will have more frequent samples, more sample size. Sampling zones will be very, very useful when you are collecting those samples from the environment. And you don't want to over collect the samples. You really want to target on where is the important risk. So the most important risk is zone 1. Depending upon the risk, you can divide it into zone 1, zone 2, zone 3, and zone 4. So zone 1 is any surfaces that come in contact with the food, and is known as food contact surfaces. If the food is exposed to the if that surface is exposed to the food, like your conveyor belt where you are processing your food, or your processing table, those are food contact surfaces and are regarded as a zone 1. Zone 2 is very close proximity to your food contact surfaces, like equipment framework, like outside of that equipment, or the control panels where the operators have to toss the control panel very frequently, or the side of a tunnel, or even the like under the table, like the same table, the up where you process your food, that is your food contact surfaces. And if you go under the table, that is zone 2. Zone 3 is remote, like a non-food contact surfaces, but are in the same processing area and could be a source of contamination to zone 1 and zone 2, like floors, walls, like drains. You may have overhead piping, forklifts, like in the same processing area, cars, doorways, cleaning utensils. All those can be designated as zone 3. And zone 4 is the non-food contact surfaces outside the ready-to-eat processing areas, like offices, like locker rooms, restrooms, hallways, cafeteria. So depending upon the risk associated with your facility, you may divide that into different zones, like zone 1, zone 2, zone 3, and zone 4. And with these different zones, you may allocate or you may collect a different number of samples, and maybe frequently also. For the low risk, you may collect less frequently. For the high risk area, you may collect more frequently. And the target microorganisms, here you can see the microbial testing, you may be looking for pathogens, but as I mentioned before, you may also look for index microorganisms. And especially, especially when you are collecting samples from the food contact surfaces, you should be collecting index or indicator microorganisms, because as you all know, pathogens are present in very few numbers, and sometimes it may go undetected. So if you detect any index organisms or indicator organisms in your food directly or food contact surfaces directly, then there is a chance that those organisms may contaminate your food, and you have to really follow corrective actions. Those are some of the sampling tools that could be used, like swabs, sponges, gauze, even contact lists, very easy to use and then collect and then see what is the prevalence of organisms in your processing environment. Floor sweeps, if you are covering much more big areas. Dust accumulation, if you feel like, a dust may be a source of contamination, or air samplers, you can see from low pressure areas to high pressure area, the movement of air, you feel like there is some leakage of the air, and air may be a potential source of contamination. This is the way. So everything tailored to the facility or the product that you are processing. Okay, and then the surrounding environment. So the sampling tools that you use must be tailored to your particular facilities. This is an example how you can composite different samples. You can use different approaches. Here, I just highlighted 3 different approaches. It doesn't mean that you have to stick with 3 different approaches. You can have a different approach also. But the important part is how you can find those target microorganisms who may be a potential source of contamination. So in approach 1, you can sample. There are examples of multiple floors. You can sample multiple floors with a single sponge, and you can use a single enrichment. That's fine. Okay, you can use another approach, like approach 2. You have different multiple sponges used for different multiple sites. Here you did a single enrichment, and then try to see if there is any microorganisms present in this sites or not. For the approach 3, you use sampling multiple sites with multiple sponges. You use 2 enrichment over here, like a multiple primary enrichment, and then you combine that, like you take a portion of those multiple primary enrichment, and then you go to the secondary enrichment. The benefit of approach 3 is if you have any contaminant related to each site, you can actually go back and trace it, because you need to save your multiple primary enrichment. You refrigerate that product, and if you get a hit on your final enrichment, final product, composite product, then you go back and then check individual enrichment to see where are those organisms from, where is the harbour site. So the approach 3 may be good, but again, you can see here, when you go to the approach 3, you need more resources, more, more budget, more funding. So depending upon how much you can spend, you can use a different approach. Sampling frequency depends upon, if you need a third-party certification, for example, safe quality food. Most of the food industries here in US or even in Europe, as I understand, will require SQF certification. And one of the important requirements of the SQF certification is environmental monitoring program. So depending upon the risk of your product, if you are working on a low-risk product, like a very low frequently, even like 1 a year or as needed will be enough. If you are working on high-risk product, you have to have at least once a quarter. It doesn't mean you have to stick in this once a quarter. We have several clientele that does, like a weekly, like a bi-weekly or monthly. So depending upon the risk, of your product, you can change the frequency. But validation and verification is very much important. You really need to verify the cleaning and sanitation is working properly. And comes the sample numbers. Again, sample numbers depends upon the size of your facility. If the facility is larger, you need larger sample size. If you number of samples, if your facility is smaller, your sample size or number of samples will be much more less. And then the sampling timing. You really need to, if you are using that environmental monitoring program to verify your cleaning sanitation practices, then you should probably focus on, collecting samples before and during production. Sample number, as I discussed, depends upon the facility size. As a normal rule, if your facility size, like a normal rule, you collect around 1 samples for every 1,000 square foot. So depending on if you have 50,000 square foot facility, you may be collecting around 50 samples, or maybe 30 to 50 samples. It depends upon, so. Again, the condition of the facility. If the facility is old, there may be a potential leak of surrounding areas of your facility, potential contaminant sites, your sample number may be different. And budget, if you have really good budget and you don't want to take any risk of recall or any risk, then you probably put more investment on aggressively trying to find any potential contaminant. So all depends upon the product, the risk, the surrounding environment, and the manpower, and then the budget associated with you or with your facilities. When to collect samples? Again, the intention. Why are you collecting samples? Are you going to verify your production practices, your cleaning sanitation technique, or you are aggressively looking for any harbor sites? Okay, depending upon your intention, your sample collection timing may be different. If you are collecting samples to evaluate cleaning and sanitizing effectiveness, you really need to make sure that you collect samples after cleaning, but before sanitizing, prior to start of operation, and after cleaning and sanitizing. When you are collecting samples after you sanitize, this is a really tricky time, okay? When you collect samples after you sanitize, you really need to have those timelines, because there may be still residual sanitizers that are present on those food contact surfaces or non-food contact surfaces and may interfere with the microbial count. So you really probably have to wait maybe 1 hour, 2 hours, 3 hours, depending upon the type of sanitizers that you are using. Okay, if you are collecting during the production operation, then you really need to keep in mind no less than 3 hours. after the start of the production. You can collect at the end of the run or end of the operation shift, or at shutdown, like before cleaning, like when you are trying to really evaluate if there is any potential contaminant during the production operation. You also have some special event. You may change the equipment, or you may have a, like a flooding in that area, like outside, not inside the facility. Like you have some constructions going on. You may get new process area or equipment line or major maintenance. Like then you really need to focus on, like where are those sites, where are the potential risks, and where these microorganisms may be a source. So depending upon the events, depending upon your intention, your timing of sample collection may be different. Additional things that you need to consider is adverse event. That's I just discussed about that. Some of the example of adverse event is you may have a roof leak. Okay, sometimes things can happen. You may have a flow drain backup, and when you have a flow drain backup, you've got all those organisms from the drain back into your facility. So you really need to aggressively find if there is any organism still left after your sanitation and cleaning process. So that's another important part. And construction of a new line installation. So those are some of the adverse events that can happen. This is an example. This is an example of tracking results. Those numbers that are presented here are just hypothetical. Like you divided your facility into 3 different areas, areas A, B, C. You are collecting 25 samples every week. You see, like some samples actually got positive. Like you got one positive in area A. In area B, week 2, you have other samples that got positive. Week 3, you have actually positive in area A and B. But those numbers that when you collect periodically can be used as a baseline to identify a potential source of contamination. Here, like your facility, you divide your facility into different grade lines. Very, very effective. Like if you are really collecting samples from different areas, a grade line that indicates different area, very, very useful. And then you collect samples from different grades. You have a positive hit week 1 in the area A, and then in week 2, you have 2 samples that are positive, very close grades. You see there, maybe a source of contamination is over there. And then in week 3, you have another source of contamination there surrounding that particular area. Okay, and then you compare, and in week 4, your contamination actually moved to the area C. Then we actually compare to your physical map, like what's going on over here. Like you have this source of contamination, like all those organisms, you are getting positive over here in one area, and then you saw the movement of those microorganisms in a different area. Then you find that there is a structural column surrounded by a gap over here. And then your employee flow, actually employees, with the movement of employees from that area into the area C. That means that there is a potential source of contamination in that area, which actually carries into your area C. And when you went over there to physically investigate what's going on, you find that there was a leak. A leak in that structural column. There's a water leak, moisture leak that's coming up between the column and then actually draining and leaking through the floor. And then the employees who are walking over there carry those organisms and then contaminate the area C. So this is very, very useful. Whenever you do those monitoring program, you should grade and divide your facility different areas. It will be very easy for you. What happens when you actually get a positive hit? Okay, I'm not going over all those charts. This is from the FDA Listeria guidance. When you have a positive hit, it depends upon if that sampling area is a food contact surface or non-food contact surfaces. If you have a positive of Listeria species, I'm not talking about Listeria monocytogenes. Okay, if you have a positive about the index organism Listeria species in the non-food contact surfaces, the first thing that you need to do is clean and sanitize. The area that's positive occur, and then again retest. You really need to go back and then see what is the product that you are dealing with. Does that support Listeria monocytogenes growth or not? And depending upon what the product characteristics is, you collect the positive or negative, you follow this flowchart. So there is a very clear guidelines on what you can actually do when you have a positive from a non-food contact surfaces. But when you have this Listeria species positive in a food contact surfaces, you need to do more than that. The important part is there is a potential of contamination in your food. So you really need to consider holding that product and taste the product also. Like you really need to clean and sanitize the line, and they retest the food contact surfaces during the production side scale. And the important part is to do a comprehensive investigation. Where are those niches? Where are those harbor sites? Where these organisms came from? You need to find that out. So in summary, hygienic zoning can minimize sanitation issues in a facility. Like you can identify areas based on risk of contamination, and you can manage traffic flow between those areas, and that would reduce any potential source of contamination. Environmental monitoring program is a verification tool for sanitation. You can expect to periodically find the target microorganisms. So when you are working in an environmental monitoring program, you really need to tailor that program to your facility, the type of the food that you are dealing with, and the environment itself. This is the LSU AgCenter Produce Safety Resources. So if you go to our LSU AgCenter Food Safety site, you will get a lot of resources. We work very closely with our stakeholders. There are a lot of publications out over there, extension publications, research publications. We still have to update, but feel free to visit the site. If you need any information, shoot me an email and then I will help you out. These are my current research focus. I didn't discuss about my research area before, but I work very closely with the USDA and FDA and state stakeholder here, Department of Agriculture, Department of Health, and food industries. And my area, my focus area, right now, current research focus area is on UVC light treatment. We have done a lot of UVC light treatment, looking on different matrices associated with different microorganisms, even in an agricultural setting. My interest is also on microbial diet, because this is a really important area that FDA and USDA are looking for, collecting data of microbial survival, microbial survival and how these microorganisms behave with all those different surface characteristics and all those different production settings. We also work on natural antimicrobials. One of the areas that I'm right now focusing is on pecan extract. So we are working in collaboration with the University of Poland. We are extracting Poland. My graduate student visited Poland, not the last year before that. And another graduate student was supposed to visit over there. I actually, I welcome two faculties from University of Poland to work in my lab. So we have a project going on with natural antimicrobials with University of Poland. We are working on antimicrobial treatments on fresh produce. We work with chlorine dioxide, gaseous chlorine dioxide. And the important part we are looking actually for is how the sanitizers will be effective in different conditions, like dry treatment, like wet conditions, like how long you really need to wait, what happens with these organisms after you treat, and what will be the residual sanitizers that are present on the produce that will be useful when you actually pack those produce and then sell it in the market. We also focus our research on agricultural water treatments, one of the really important areas here in US, because water is the important source of contaminants. And as Dr. Ariana mentioned in his presentation, water is an important source of contaminants. If you are growing lettuce, like if you are growing other crops that are consumed raw, you really need to make sure that the water that you use for irrigation is of high quality. So we are really focusing on how to minimize the risk associated with agricultural water. Another research area is on hydroponic production. Like in hydroponic production, like we use water-soluble nutrients, and those water-soluble nutrients are also good medium for the growth of microorganisms. So we are trying ways to minimize the microbial risk, but at the same time, we want to maintain the quality and then the production of the crops that are used in hydroponic system. We are really great on social media, guys. If you want to follow our Facebook and Instagram, LSU AgCenter, Food Safety, Twitter, we do update a lot of information. So please follow this like social media site. Any information, any other things that you need, shoot me an email, text us from the social media, and then we will be able to get back to you. And I will be happy to answer any questions if you have. And let me, okay. So let me stop sharing. That way I can actually. Good? You guys see me?
Yes, we see
Awesome. Awesome. Is there any questions that I can answer?
There is a question on the chat. Just a second. So in which food sector are the biggest problem with hygiene in the US?
That's a really fantastic question. Like whenever we have workers or like people working directly with the food materials, then we have a really high problem of hygiene. Especially like with the berries. Like when you talk about strawberries, blueberries, those are picked by hand. Really need to focus on the controlling the health and sanitation of those workers. We actually have offered one program. We also do a lot of extension outreach activities and train our growers. One of the training that we offer is worker health, hygiene, and sanitation program. So my graduate students are bilingual, and a lot of workers here in Louisiana, they speak Spanish also. So we go to the farm, we go to the production facilities and actually train and use hands-on activities to train those workers on increasing the knowledge about food safety, on risk associated with that processing line, and how to minimize the risk. So great question. The important part is whenever you have more interaction of the workers with the product that are ready to eat, those products that are consumed raw, that's where we have the hygienic problem. We were discussing about COVID-19 just before, Dr. Ariana. We had a huge problem here with the crawfish. Like last year, we have to actually close a lot of crawfish industries because we have a lot of workers that work in the crawfish that actually peel out the crawfish and then separate the tails. But with the COVID, we have big outbreak related to COVID with the workers, and many crawfish processing facilities were shut down. Any questions? Anything else? I'm trying to make sure that I'm not taking more time, because we have a definite amount of time, and I'm looking at the clock. So any questions that I can answer, I will be glad to answer.
There are no more questions in the chat.
You has answered several different questions. That's great. Any other questions that there's over there?
Okay. Thank you, Dr. Adhikari. Thank you so very much. We are just on time for our next speaker. So a wonderful presentation. Thank you again. Our next presentation is by Dr. Venitsu. Dr. Venitsu is an assistant professor and consumer food safety specialist with the School of Nutrition and Food Sciences. Her research focuses on using reality-based research methods to enhance microbial safety of fresh produce, as well as understanding the survival and transmission of pathogenic bacteria and viruses in various public settings. She is also passionate about food safety education for different targeted audiences. Her topic today is about food safety communication with general public. Dr. Sue.
Thank you, Dr. Ariana.
Thank you.
Can everybody hear me okay?
Yes.
Okay. Everybody can see my slides?
Yes.
Okay, perfect. Thank you. It's always good to check. Good morning and good afternoon too. So I'm the third speaker of this morning, and I like to switch gears because I know Dr. Achut and Dr. Kai talked a lot about the basic microbiology and some of the practical research. I like to talk about the food safety communication with general public. Just a little bit introduction on what do I do. My lab conducts practical research focusing on multiple foodborne pathogens. We work with Listeria monocytogenes, E. coli 0157H7, and Salmonella typhimurium. We also have a project working with human norovirus. So my lab is very diverse, and we have a lot of fun projects going on. I like to show those two pictures. I love them. They are E. coli 0157H7, but both they were treated by the pulse light. One of the light treatment, they are stronger than typical UV. So as you can tell, I don't know how clear it's on your screen, but on the top left, here you can see the edges of the cell all flat out due to the light treatment, the sudden water evaporation from the cell that how it kills the bacteria. But when you add water to the treatment, put it under the light treatment, then you don't see that type of death methods. You see a lot of holes formed on the cells. So I think those pictures kind of cool to share. What do I also do? I also, as Dr. Adhikari talked about, is we have both research and extension work going on in my group, and I have sometimes in the lab, but also I have times in the communities and teaching people about food safety. So I have a extension component to my job, which I love it because those research and extension actually they really go together. So for all my projects, as Dr. Kai said, I'm a assistant professor, but also a consumer food safety specialist. So my research and my extension projects ties together. All my projects are going through the same type of logic flow. So we have the needs assessment, then we have the educational material development. After the development, we figure out the best channel for the communication to disseminate the information. After the information is done, we follow up with the evaluation. This evaluation piece normally lasts 3 to 5 years. So we don't have all the data yet for this one, but I would like to highlight some of my project from my group, so you kind of can see how the research and the extension tie together in my group. First project I like to share is the pregnant women and food safety. When I started with started at Louisiana State, first of all, I think I'm gonna work with general public, but also I want to specifically work with our higher risk populations. High risk populations from the food safety standpoint, we're talking about pregnant women, very young children with age of 5 or less. The elderly, we're talking about 65 or above, and immunocompromised groups. So try to identify their needs and their needs for food safety education and deliver the effective program. That was my priority. So this project focused on pregnant women. And we have conducted needs assessment, and we evaluate the awareness of listeria and high-risk food consumption behavior among pregnant women in Louisiana. We also look at the overall pregnant women's confidence and perceptions on practices related to food safety study. This also focuses pregnant women in Louisiana. What we have identified in those two studies is pregnant women self-report that they're aware of listeria, the risk of listeria monocytogenes, and they know some of the high-risk food. But when it comes to behavior, they don't necessarily adopt the behaviors that avoid those risks and mitigate the risk of catching listeria monocytogenes. So we identify there's knowledge and behavior gap over there. So we developed some of the educational material. What I'm showing here is one of the fact sheets we developed, talking about for moms to be. There are two types of things we worry about, right? It's listeria monocytogenes and Toxoplasma gondii. One is the bacteria, one is the parasite. And so we answered a lot of questions for people. What can you do? What can you not do? Some people wonder, do I get to keep my cat because of the Toxoplasma concern? And Toxoplasma not only come from the cat litters, they also come from undercooked meats. So with a lot of the information here and how to cook, how to make sure your food is done, is properly cooked. So to go with this, we also have the magnet on the right side. It's a magnet about kind of like action plans, how to reduce the risk of the listeriosis and how to reduce the risk of catching Toxoplasma. So this you can put on your fridge during pregnancy, and it kind of remind you of what you should be doing. So those are the some of the educational materials we developed, and our communication channel is through the Baton Rouge Women's Hospital. This is our collaborator in this project. They have the Baby Grant events. It's a biannual event, and people from the urban Baton Rouge urban area and suburban areas all come to this event, and that's a Baton Rouge Women's is the biggest women's hospital here, and if I remember correctly, it's number 17 at the national level. So the education power of this of the hospital is amazing. So we use their platform and we distribute our materials there and talking to pregnant women and try to raise awareness of avoid Listeria monocytogenes and toxoplasma. That was one of our projects. And our second project, speaking of targeting on the young children, we looked at the animal contact in public settings. So if you look at this, I have the table here, have some stats. There are all the people getting sick from contacting with animals and the public settings. Public settings, normally we're talking about educational state fair, open farm, county farm, petting zoos, like all those. And the backyard flocks has been causing problems every year almost in the past few years, and a large number of people getting sick just because of the lifestyle. People try to raise chicken as a interest, as a way of I can collect my own egg. Actually, the kids treat those chicken as pets, and they don't necessarily wash their hands after they handle them. So it has thousands of people getting sick from the backyard chicken. As a picture on the right side, you can see that the kids at all those fairs are contact with animals, and they move from station to station. But the hand washing station is normally at the end and the exit of the fair. And we try to see how many of them actually used the hand washing facilities, hand washing intent, and at least try to wash their hands. So we had the needs assessment, looking at the animal contact in public settings, risk awareness of enteric pathogens, and hand hygiene behaviors. The study is like 2 parts. So we survey the parents about how do you kind of look over your kids and ask them to remind them to wash their hands after they contact with the animals. How often, how fast do they wash their hands before after they get in contact with animals, or do they have food or beverage consumption during this time at the fair, and all sorts of risking behavior like touch your face, touch your lips, eating and drinking, that type of high risk behaviors. And we also observed the hand washing intent, how many people actually walk to the hand washing, waiting in line, and try to at least have the intent to have the hand hygiene behavior. So what we have found out from this study is published is the people's self-reported awareness is high, but they don't necessarily relate the animal with foodborne pathogens. They don't necessarily relate the cute animals they just pet could give them E. coli L157 or give them Campylobacter or other type of the pathogens. They don't necessarily relate. They know the pathogens, and they don't necessarily relate the behavior with the pathogen. And the hand hygiene behavior, and between the awareness and the hand hygiene behavior, there is a big gap too. So to address that, we have developed some of the educational material. This fact sheet is called Share the Knowledge, Not the Illness. So kind of list out the risk of animal contact in public settings, and how do you try to prevent the illness. The channel we distribute is through the AgMagic, and we work with Department of Communication. So we have the fact sheet in every teacher's packet, because before the setup of the AgMagic is we have the animals and all the displays set up on campus, and all the schools, they're gonna come over, teachers gonna bring the kids over by a set schedule. Before they came over, we have the package for the teachers, and so teachers could have the information read to the kids before they come, or teach them in the classroom before they come, and emphasize those hand washing behaviors during and right after the tour. So this we found is to be the most effective distribution channel for us in this specific topic. So for the third project I want to share, is kind of switching gears, is that food safety for home gardeners. We have a lot of home gardeners, and they grow crops, grow produce, and then they probably have too much for self-consumption. They would kind of preserve them or bring them to the farmers market or give them to the friends and family. So those home gardeners, but we also have a lot of the flooding events. Well, in Louisiana, the rain and the flooding is not unusual, right? So this is a picture of us taken in 2016 with all the fields has been flooded. And we got a lot of questions from the home gardeners. It's about what about my crop? Can I save my produce? What can I save? What I have to discard? What is safe? What is not? So when you look at the FDA recommendations, FDA said if you have the edible portion of the produce came in contact with the flood water, you have to discard it because that's considered adulteration. But if you have produce grow near the area that has a flood, which means your edible portion of the product is not submerged underwater, you need to analyze a case by case. But a lot of times it's harder to analyze case by case because people don't necessarily have the resources. So we have conducted study to evaluate the microbial safety and quality of Louisiana strawberry after flooding. So the picture on the left is our group of fully submerged group with our strawberries. With the flood water is used. We use cow manures stirred in the water and spiked with generic E. coli to simulate the flooding. And we have another group of the plants that's not submerged, but the flood water kind of passed in the plants, but did not, edible portion of the plants did not really submerge with the flood water. So, and of course we have a control group. So those are our studies published, and we also collaborate with Virginia Tech. They have conducted a much bigger scale of simulated flooding research, and they set up their fields like this, and they flood the fields with water and trying to get samples of the soils and get samples, try to get samples of the water, kind of to see try to find out the pattern of the risk. So we can kind of design our risk management plans after the flooding. So one of the educational materials we developed is one of these fact sheets called Microbial Safety of Fresh Produce in Home Gardens After Flooding. And kind of, this is just the front page of it. It's, I think it's 3-page long. This one is a long fact sheet. Also have tells you how long you need to wait before you're going back. What is a safe buffer zone? Why do you have to have buffer zone for you harvest your produce? All sorts of information on here. And I don't know if you can see this on the right side. If I click play, this is one clip of our e-learning modules, it's multiple components. The first component is kind of walk people through the risk of flooding, because sometimes people think it's just rainwater, right? But it's not necessarily rainwater. It can bring in chemical and biological hazards. So this is one of the animation from our module, kind of tell people, okay, your field being flooded. Now you have this beautiful lettuce and being flooded. We have voiceover from the module. I just took it out. I don't know if y'all can see this. Half of the time people say they can't see. So now after the water recedes, if you zoom into the surface of the lettuce, you can see the bacteria attaching on the top. Normally people don't get to see the bacteria because it's too small to be seen by naked eyes, but I wanted to show people how bacteria can get attached to the surface of produce through the animation. Okay, so our distribution channel for this type of information is, first of all, we distribute it through our collaborators, Idaho and the Virginia Tech, and we also locally we disseminate through the Louisiana Fruit and Vegetable Grower Association, passing our information along and kind of help address some of their concerns. So last but not least, the most recent project we had the food safety information on social media during COVID-19. Earlier, I saw there was questions in the chat when Dr. Adhikari was presenting, asking about American consumers' perceptions about the food safety. I would say people's perceptions about the safety and the risk really changes a lot, depends on what they see over the media. In the past few years, we think it's a more mass media with affecting people's view about the risk and the safety. Now it's we see a lot about the social media impact on people's perception about the risk. So this study has been done on YouTube content for food safety related information during COVID-19, and it was just accepted by Journal of Food Protection. And we're looking at basically what type of information, what type of videos had very high views, very high comments. We analyze those views, analyze those comments, and analyze their content. Basically, we code the when certain things when the video mention about they have the government agency information cited, or they have the health professionals serving as guests or hosts, or they have some sort of produce washing tips recommended, hand washing tips recommended, how to do grocery shopping, how to do takeout foods, and all of those behaviors are coded. Then we look at them in a numeral fashion, but they're descriptive study. So from this study, we found that there are a lot of misinformation out there. On top of my head, I think one is because one suggests to leave your groceries outside for 3 days, because one study published by the New England Journal of Medicine, I think published in February, about how SARS-CoV-2 virus survive on the surfaces, can survive on cardboard box for 3 days or something. So people just kind of think if you leave your groceries outside for 3 days, that can be virus free, but at the same time that's violating the food safety practices. So coronavirus may not kill you, but the foodborne pathogens may get you, right? So some stuff like that, and people suggest putting the fresh produce through dishwasher, which is just absurd. Some of those behaviors, and followers of the video and the comments, we kind of see how people's taking on those informations. So we developed our own educational materials. We had a series of fact sheets. So this is just one front page of one fact sheet. This was talking for the public health emergency response for retail store managers, and we kind of help them identify like what is your high traffic area, what is your low traffic area, how should you set up your high traffic area, how to space out people. And we also have it for like the local seafood processors and fishermen, because this is a collaboration between Louisiana State Grant. So it's I think we have multiple fact sheets and translate in Spanish, Vietnamese, and Chinese. It's based on the local populations. And on the second here, we have that's one of our posters. Basically tell people to spread out. And this looks not that crazily new these days, but like this was out by March 2020. So before a lot of other agencies send it out. So this fact sheets and this posters, and I think we have 6 posters total, have been used by New Orleans City Council for distribution. And all the fact sheets has been used by Louisiana Department of Health for their distribution to all the establishments in the state. And we have the video here talking about the for consumers, talking about how to use the disinfectant correctly, because people normally don't read the label. I don't think we have time to show the video, but if you're interested, I can send the link. It's on YouTube. It's a minute long, so okay. So our distribution channel, talk about all those cool materials we developed, is through Department of Health, and through State Grant, through New Orleans City Council. We also send emails specific to our industry contact with over 1,200 contacts. And we also put on social media. So for the video I just showed you earlier, it itself was over 2 million reach on the social media. So that's our distribution channel for this project. So those are some of the project highlights for my group, and I like to just kind of talking a little bit about the communication, food safety communication for various of audience. I think the biggest challenge is the different audiences. You have to kind of use different approach. For scientists, all they care about is statistically significant, right? You show them the data, the bar graph, the pie graph, whatever you're showing them. If it's not significantly different, we're looking at p-value, right? If the p-value is not significant, you basically did not present anything, right? So they care about the significant difference. General public doesn't care too much about that. They care about immediate practical impact. What can I use? What can I do with your information? You just give me 15 minutes on presentation. What can I use the information for? That they're like very short attention span, and they wanted to put the stuff in use right away, right? For businesses, also work with a lot of local companies. They care about regulations, care about lawsuit, care about the profits and costs. So you can say all kinds of risk about the product, whatever. If you say, okay, this is regulated and you need to comply, so they would more likely to adopt, to listen to what you have talking about. So I think I have some time to kind of give you some scenarios, just 2 scenarios about the, kind of show you how the same information can look so different in different scenarios, depends on the target audience. So now I need you to think as part of, you're part of this scenario. So scenario number 1, we're at the International Association for Food Protection annual conference. So everybody who sits here are food microbiologists or food scientists at least. So we're talking about those people are your peer scientists. Okay, so this is a presentation I give, and that setting is survival and inactivation of Salmonella enterica in long-term stationary phase or LTS phase on food contact surfaces. So talking about long-term, what is long-term stationary phase, LTS? We heard about the lag phase, the log phase, the stationary phase, right? That's what, when we do microbiology study, we focus on those 3. Most times the stationary phase. But long-term stationary phase is like after the death phase, you have the bacteria reach this stable kind of long-term stationary. They can survive being there for months. Okay, just not change. So they're not dead all the way to the 0 count. So they get to their long-term stationary phase. So to communicate with the peer scientists, my intention is not to present our findings here. I'm just trying to talk about the communication. You don't need Nancy background information. You don't need to tell people, oh, what is bacteria, what is the log phase, stationary phase. People know about those, right? Because they're your peer scientists. And we need to identify the research gap and propose your hypothesis. So our gap, we find the gap is here. All the study were done, most study were done on the stationary phase or log phase. There is nothing done on long-term stationary phase. But when you think about it, in the food facility, processing facility, if the bacteria been there, they've been there for a long time, right? So this phase, where LTS phase is kind of, the information on that is lacking. So we want to study the difference between this phase and the stationary phase, what we normally study. So that's our hypothesis. We think when bacteria gets to this phase, LTS phase, they're going to be more resistant. So this is one of the graphs of our study. Again, ignore the content about this, but what I really want to show you is, let's look at the last 3 bars here. Okay, so high inoculation level 10 to the 6 CFU per square centimeters on the plastic cutting boards on the plastic, and we have the 24 hours after the soil of the surface, then we start to disinfect it. And then we have the log phase, stationary phase, and LTS phase. And you can tell the LTS phase is has a significantly higher survival population of Salmonella on the surface if you don't clean it right away, right? When you have, especially when you have the high contamination level. So the summary of the scenario 1 is LTS cells shows higher survival population than the stationary and log phase. And the highest survival population, about 5.8 log, was recorded for laminate surface. That previous I showed the graph with the plastic surface. We also have stainless steel surface and laminate surfaces. So communication to the peer scientists, of course, we want to explain in details about the significance of the data, and they can tolerate the boring, right? Because I can tell them look into each of the groups here, low contamination, high contamination. I can look into each bars. They can tolerate it because they want the information and they're interested in information. They don't think it's boring at all. But you cannot do that for the general public. So our scenario number two is for the community workshop. Those are the people who are volunteers during the natural disasters, which means they volunteer to cook for the communities after the natural disasters. None of them have the professional trainings on food science, food safety, nor probably not even cooking, right? So those are the groups of people we're teaching in the community in this scenario. And I normally use activities, just kind of ask them to, how do you clean your kitchen countertop? And what type of product do you use? How many times do you clean? Do you read the instructions label? How long do you wait before you clean it? So with the hands-on activities is more effective when it comes to general public, and they will remember, especially when they did it and they did it wrong. That's where they would remember it. So those are the activities and the summaries. I cut it pretty short for the scenarios, but you'll get my points in a little bit. Talking about the cross-contamination, how to prevent cross-contamination. One of them being like you have to, if handle raw meat and poultry, if there is spill, you need to clean and sanitize right away, right? And use detergent according to the manufacturer instruction. So for the communication to the general public, I want to tell them exactly what I just taught. So recap and bring home messages. This is important when you teach general public. But if you really think about this, scenario number one and scenario number two, they sounded like very different presentations, but they are actually the same presentations. If you have potential contamination of your surfaces, you need to clean and sanitize them right away, and you need to use the detergent according to the manufacturer instruction. It's exactly the same message, but you may not even recognize them as the same message because they look so different. But again, they look so different because we're facing different target audiences, right? If you teach the peer scientists about how do you clean the surface, how do you sanitize the surface, they're not interested in that. They knew that already. But if you bring your graphs and all those data to the general public, they're gonna lose their interest. They're not gonna listen to your whole presentation. So sometimes same message may look very different, depends on your audiences. So I like to give acknowledgement to our funding agencies, as well as our Department of Communication helped me with all the communicating materials, and all my students and my collaborator with Louisiana State Grant. This is our safe food specialist. And that's my last slide. I'd like to say, when it comes to food safety communication, the tools have changed. We changed from this phone to the iPhone. Now, this is the kind of old iPhone. We have fancier iPhone now. But it's not just a tool change. The content has to change, the way of delivery has to change, and the take-home message has to change. Depends on your audiences. Thank you.
Dr. We do have a couple of questions on the chat.
Okay.
How do Louisiana government authorities get into hygiene issues, and how is it at the federal level? Is the issue of education in this area addressed only at the local level?
All of the study we have done is trying to address local level needs, but our government agencies, you're talking about Louisiana Department of Health, and they try to reinforce their basically the food code for Louisiana is Title 51 across the board. But we do see some variations of the practices, depends on the businesses. But what we're trying to do is kind of develop the customized education for them, even though they have to comply with the standardized regulation. The education needs to be customized.
Thank you. And we do have another question. Is there any data available on animal contact in domestic settings, by which I mean getting a disease from one's own pet? People often share food with their pets and are much less careful. Would you say the risk of catching disease from our own pets is higher due to reckless behavior or rather negligible due to better hygiene standards?
That is really good question. Actually, there are multiple studies out there about the animal contact. When you're talking about your own pets and share food with your own pets, it's not necessarily about your pets. It's about the pet food and stuff you are feeding you, and you share the kitchen. Use your kitchen microwave, your kitchen countertop to prepare those foods, or you contact the dog food. You did not necessarily wash your hands, right? So those cross-contamination practices in your kitchen that actually have a higher risk as well. And earlier we talked about the kids thinking the backyard chicken as their pets, right? They pet them, but like again, backyard frogs has been causing outbreaks almost every year in the past few years, and the number we are seeing are really high. This is a lifestyle change, and people really need to watch their hand hygiene behavior needs to be improved as well.
Thank you. I do not see any other questions, but Dr. Su, if you could please send us the link to the video so that we make sure we post it later on for everyone to see.
Will do. Oh, also, can I just say one more thing about the pet food?
Oh, yes.
A lot of times these days, people, I'm recently found interesting in pet food. That's why I'm like, I want to say a little bit more. People try to fit a raw diet to their food as if raw diet is better, thinking humans, we want to go back to our ancestors, right? We want to eat as raw as possible. Now with pets, people have the same idea. I'm going to fit my pet raw dogs, like cats, whatever, raw food. But like, again, we had consulted with the vet, and those raw foods may not necessarily provide better nutrients to the pets, but that's not my area. My area is when FDA conducted a study to sample the dry pet food, semi-dry pet food, and wet pet food, and raw pet food. So you are the raw pet foods, the bacteria content is much higher than the dry food. Dry pet foods. If you really want to feed your pet with raw pet diet, you have to really do some good job to prevent cross-contamination, because I assume you're doing that in your kitchen. So you really need to prevent cross-contamination. Some people thaw because they come in blocks, they're frozen, right? Some people thaw them in their own microwave, and then later they put their own human food in the microwave to heat it up. So those are type of behaviors we really need to watch out. Whatever you are switching practices, you need to look out for your hygiene behaviors as well.
Thank you. If we do not have any more questions, thank you very much, Dr. Shu, for presenting. It was amazing. We will be moving forward to the roundtable discussion, which will be led by Dr. Miroslav.
Yes. Hello to everyone. Do you hear me?
Yes, we do.
Perfect. Thank you very much. So I have a little presentation for you. So I will upload now. Yeah. And I have a request for Ivana. Can you add Dan Falta and Ivo Ondrašek as speakers? Is it possible?
Yes, it is. Give me a second.
Perfect. They could say a few sentences at the end, I think, and I saw that they are connected. So perfect.
They should both be having the privileges now.
Thank you very much. So we have very good time because we have 1 minute to our roundtable discussion section. So it's a perfect time schedule. And I'm very pleased that the Food Symposium has fulfilled what we probably hoped for. So, and much more, I think. And the food safety blog was perfect. We have many questions in chat, I see. And if we can continue for some time, for a few minutes, we can deal and say something about whole week. So if we are now, yes, 16. So the Thursday as program was very, I think, very important for us, for Department of Food Technology and for Faculty of Horticulture. I think we have great potential for joint course or cooperation involving students. I saw many students here, and creation of new research and multidisciplinary teams, commercial cooperation, and so on. So this is the, I think, preview of my roundtable discussion. And I have to say great thanks to all. So we can create and continue. So if we can see this, because from the title of our final block comes the question, opportunities and threats in the key aspects of food system. So if the food security, safety, and nutrition are compatible. So I think, of course, and it is at our great event, the Food Symposium, that you can see how much. So I see here the conclusion and some possibilities of cooperation in nutrition. We had in Wednesday, and food production and quality section, food technology, we had in Thursday, and now in Friday we had the final block of food safety. So you can see that there is compatible questions, compatible in research, in education, and together with Mendel and with LSU. So if we continue in my slide, next slide, yes, research is essential for the control, management, and sustainability of these systems, and teaching, because we are university and our activities go for teaching, for education too, is aimed to create and specific workers, experienced workers, as a basic tool for food security, food safety, and nutrition. So this is my final few words to this theme and to this program, and I hoped for the cooperation, and we have many contacts, so we can continue. So if we can see this last slide. So many thanks for your attention, for your cooperation, and I look forward to cooperation, do cooperation with you, with LSU. So this is my contribution, and if you have something to chat or something to say, so you can ask. Could I ask Ivana for some little words?
Of course, I wanted to thank all of our colleagues from Mendel University and from LSU. I really liked the discussions that were going on in chat, and I can already see how some potentially cooperative new teams have formed. At this point, we have established at least 2 or 3 groups that already are scheduling a follow-up meeting, and we would like to encourage you. If you have any interest, please do let us know. In a chat is fine. There are some faculty members that may like to cooperate with student programs. There may be some that are focused very heavily on research. Dr. Shu, that was our last presenter, actually is more of the outreach and communication to the public and research focused. So we really would like to ask you to either put some questions in a chat or maybe at least give us your interest. What is the main interest? What topics would you like to cooperate? And we will be following up in the upcoming weeks and trying to create linkages and seeing if there is a way for you to cooperate. For the students that are with us, I would really like you to let us know what your interests are. There are programs between our universities that allow students from Mendel to visit LSU or even spend a full semester at LSU. We already have a couple of direct results, and we have an LSU student that is interested in a full semester at Mendel University. We will be processing that. Hopefully the COVID era will end soon for all of us, and the traveling will be a possibility again. I also wanted to let you know that a couple of other results came directly already from this symposium. First of all, we have decided that we would really like to repeat this symposium and make it yearly between LSU and Mendel University, and we will be working on next year giving an opportunity to students to present their work as well. Whether it's part of your thesis or whether you are doing some other research or work, we would like to include you. So if there is anybody who is interested, please let us know, reach out. Also for faculty members, if you are interested in helping us to organize this event next year or participate in this event next year, let us know, reach out. We will be creating a team to organize this and reflect all of your interests for the topics offered to everybody for the next year. So,
and at the end, I would like to ask the vice deans of our faculties of Mendelu. First, I will give the chance to Vice Dean Ondrášek. Is Ivoš here for any some positive words to the end?
Yes, I'm here. Thank you very much. My best greetings to all in this afternoon. I am alone here in our faculty, but I can express on behalf of our colleagues, especially from the Department of Post-Harvest Technologies, our strong interest to continue in some different ways of cooperation. And I have seen some emails from yesterday evening, from Ivana and from another person in this group. So I think everything is set it, and we can continue in our exact communication regarding our goals and deals, as well as another departments of Professor Pokruda, vegetable science, and our laboratory analysis departments in our faculty. So that's my very short sentence and my very short greetings, and maybe my personal greetings. It was very nice to see all of you, and I am more than happy how our activities continue and bilateral interest still exists between our faculties or institutions. So I would like to continue with you in all activities and interests, and I wish you all the best in future and normal world very soon. And I'm looking forward to see you in our place, or we will visit you in your place with some interested group of cooperation. So thank you very much to all, and symposium was perfectly focused on the modern topics and sciences, and was better than nice. Thank you very much.
Thank you very much, Ivo. And in the end, I would like to ask Mr. Vice Dean Dan Falta for short greeting and termination of this section, if he is here.
Thank you very much, Miro. I'm very glad, and thank you for the opportunity to say a few words in the last day roundtable discussion, and in connection with the topic of food, the beginning of our cooperation in 2015 were nicely pointed out in the first presentation of you on Thursday. So there was some very nice pictures where the food, Mendel and LSU began. Personally, I think that the topic of food was beautifully connected to the several workplaces, faculties, universities and departments of various specialization, and it offered a lot of other options. From the position of my vice dean position, it was very also easy to me to identify the connection between the LSU and the Mendel directly on food, because we have to eat every day, and this is our very common, let's say, topic. Thank you again for all the participants. I'm looking forward to summer school. I hope it will happen soon. And of course, we already began to find out a leading topic for the next year, maybe. And thank you again. I wish you good health, and see you soon here in Brno, in LSU, or online on the other conference. Thank you again.
Thank you very much for beautiful words in the end. And is anybody here who want to something say? If not, I think this is the end of the food symposium week, and thank you for your attention, and I look forward to hear you, see you, and to cooperate with you. So goodbye, and stay healthy, and best weekend.